Greetings everyone, my name is Aisha Ahmed and the enumeration methods for Salmonella in E. coli. This article was taken from the Journal of Applied Microbiology, also available on eCentennial. Well, till this article was published, till this study was done, the most probable number method was used for the enumeration, which is a triple dilution method, and it is costlier and more time taking than the methods that the study used. The purpose was to develop convenient enumeration methods for the two samples in ground beef, carcass, hide, and fecal samples from the cattle to make this study more cost efficient. The methodology goes like this. We use two methods, HGMF and SPCM. HGMF stands for hydrophobic grid membrane filtration. The study used it on ground beef and carcass, whereas spiral plate count method was used on hide and fecal feed. This is kind of a comparison between the MPN method and the SPCMN HGMF method. We can see here that the results for MPN could take around 40 to 60 hours, whereas for the results for these two techniques only take around 20 to 48 hours, which is quite less. And the methodology is quite simple here. The methodology as goes is for ground beef, the spiking of 65-gram sample at final concentration at 10 to the power 0 to 10 to the power 4 CFU per gram was done and allowed to sit for 15 minutes. Then 585 ml of TSP was added in 1:10 ratio. Then 9 ml of aliquid and 15 ml of conical tube was taken containing 3 ml of PBS and then allowed to sit again for 5 to 10 minutes at room temperature. 7 ml of sample was transferred on isogrid membrane. Again, this is a grid membrane filtration. That is why we are using a lot of membrane filters. And it was filtered using FilterFlex spread filter apparatus. The membranes were then transferred on XLD TNC agar plates and incubated for 18 to 20 hours at 37 degrees. A similar method is there for carcass as well. The values are changed. The 3 ml of aliquots instead of 5 ml were taken from each pooled samples and were incubated with Salmonella typhimurium at final concentrations of 10 to the power minus 1 to 10 to the power 2 CFU per 100 centimeter square. 1 ml of the sample is then taken and put into 11 ml of PBS 280, which makes it more aqueous. 6 ml of sample was transferred on again isogrid membrane and filtered using FilterFlex spread filter, again the same as we did on ground beef. The membranes were then transferred and incubated for around 18 to 20 hours at 37 degrees. Then for hide and fecal feed, it's different. Another method that was used. 5 aliquots from each pool sample were spiked with an overnight culture of Salmonella typhimurium at final concentrations of 10 to the power minus 1 to 10 to the power 5. Each aliquot was vortexed for 3 minutes and then spiral plated in, because this is a spiral plate count method, and it was spiral plated on the same media that was used for the other method as well, and then incubated for the same time at 37 degrees. Same goes for fecal feed. Instead here 200 grams of sample was taken and 5 aliquots of 10 gram each were spiked with an overnight culture of Salmonella at final concentration of 10 to the power 1 to 10 to the power 5 CFU per gram. The concentration differs and for different samples that were taken. The same method was used again, spiked sample with dilution 1:10 ratio by adding 90 ml of TSB here. 1 ml was removed and vortexed and then allowed to settle and then plated on the XLD TNC media. The results were as follows. These are the numbers enumerated in percentage for naturally contaminated samples of these 4 samples that we have taken. For ground beef, Salmonella was 5.9% and E. coli could not be determined. The values were too little to be determined here. For carcass, it was 19 for Salmonella and 15.6% for E. coli. Hide, it was 16.5 for Salmonella typhimurium and 6% for E. coli. Then again, for fecal feed, Salmonella could not be determined, and it was highest for E. coli. And the highest percentage of E. coli was found on fecal feed, that is 22.9%. The conclusion is these two techniques are more cost efficient and time efficient and hence should be used. The significance of the study was it increased the routine testing. because it's convenient and the accuracy is it's more accurate than the MPM method used. The effectiveness of beef processing strategies was increased by this. The recommendations by the author are MPN method is not feasible, and instead these two techniques can be used for the enumeration of larger number of samples. PCR and other modes of pathogen detection can be misleading because of some background growth of microflora in it. Carcass samples should not be kept for more than 24 hours. It again misleads the results. These were the references, and I used the figure of comparison of standard MPN method with the SPCM and HGMF from the same article. Thank you so much.